Good morning, good afternoon, and good evening to everyone joining from all around the world. My name is Shirin Darashani, and I'm happy to be your host for GARDP's webinar today on overcoming challenges of tuberculosis drug discovery and development. Revives GARDP's education and outreach program, and it aims to connect and support the antimicrobial research and development community by facilitating learning, connecting people, and sharing knowledge. Our webinars are part of our educational activities. All of our webinars are recorded and they are freely available to view after the live broadcast on our website, and you can see the link here in the green bar below. You can also read our Antimicrobial Viewpoints, which is a series of articles where experts discuss various topics within their fields. On the Revive website, you can also find the Antimicrobial Encyclopedia, which is a resource section that has definitions on various important terms that you might hear also in today's webinar. And some of the terms we have in the encyclopedia are also further explained by experts in their fields. As always, we will have the presentations today and then followed by a questions and answers session. So you can submit your questions during every presentations by using the functionality that you can see here on the screen. Just please remember that if your question is for someone specific, to put their name at the beginning of the question so we can make sure to address them. And we will try to address as many questions as possible. So it's my pleasure to show you today's panel. We have Jeremy Rock, Dirk Schnappinger, and Laura Cleghorn. And our moderator for today's session is Valerie Mizrahi. So Valerie is Professor Emerita and Senior Scholar, as well as former Director of the Institute of Infectious Diseases and Molecular Medicine at the University of Cape Town. Her research focuses on aspects of the physiology and metabolism of Mycobacterium tuberculosis, relevant to tuberculosis drug discovery, drug resistance, and mycobacterial persistence. Valerie has served on numerous scientific advisory boards and review committees, and she has trained more than 85 postgraduate students and postdoctoral fellows, some of whom have moved into leadership positions in South Africa as well as abroad. So welcome, Valerie. It's my pleasure to hand over the session to you.
Thank you so much, Shireen, and good morning, good afternoon, and good evening to a diverse group of listeners at today's webinar. It's a huge privilege for me to be hosting this and moderating today's discussion. What we share in common is a passion about antimicrobial drug discovery and development. And over the last 15 years, there's been a step change in the way in which tuberculosis drug discovery has been prosecuted. And the three speakers we've got today exemplify this step change. So we really are in for a great treat. I'd like to introduce our first speaker, and this is Jeremy Rock. Jeremy is an associate professor at Rockefeller University, and he is head of the Laboratory of Host Pathogen Biology, where his laboratory studies Mycobacterium tuberculosis, MTB, as we know, the leading cause of death due to infectious disease. He applies functional and chemical genomics to study not only the way in which the bacterium interacts with the host, but to understand what the bacterial factors are that limit the efficacy of antibiotics to kill this pathogen. Jeremy's background is that he did his undergraduate at the University of California in Berkeley. He worked in the pharmaceutical industry he worked in biotech actually for a few years before moving to MIT, where he did his PhD in cell cycle regulation. And we're very glad that actually the TB bug bit him when he went to Harvard School of Public Health, where he worked with two luminaries in the field, Sarah Fortune and Eric Rubin, and did some really pioneering work they're developing, for instance, regulated gene expression systems that have totally transformed the field. So it's a great privilege to introduce Jeremy. Over to you.
Thank you very much, Val, and Thank you for the opportunity to present in this seminar series. What I'll tell you about today is it's a published piece of work, so you can go take a look, but it's sort of an emerging concept on how we think about prioritizing new targets for antibiotic discovery in TB, but of course the concepts are much more broad than that. So I'm not a drug discoverer, I'm a geneticist and a microbiologist, and the way that many biologists who study a biomedically relevant problem motivate their work is that they're going to discover new biology, and that knowledge is then going to allow the field to discover drugs that perturb that biology, and you're going to end up with a new drug. And that's great, except for one significant problem, and that is that this is really hard. This requires what's called target-based drug discovery to work, and you'll hear more about exactly what this is from Dirk in the next talk. You need to start with a target and end up with an antibiotic, and the success rate of this drug discovery modality has not been great, although I'm optimistic about the future. So in one simplistic scenario, say you find a target that you think the biology is interesting for whatever reason, you purify that, you perform an in vitro screen, identify potent inhibitors of enzyme activity, and then you dump those compounds on the bacteria, and then nothing happens. And the sort of dogma for why these inhibitors don't work, they're all sort of blame the compound conclusions. So you've got this potent inhibitor of enzyme activity that can't get into TB, or it gets in and it gets degraded, or it gets in and it gets pumped right back out. For sure, these are all very real failure modes of target-based drug discovery identified compounds. But what we reasoned a number of years ago is that this certainly can't be the whole story, because it's ignoring the other side of the coin, and that's the actual targets of the compounds themselves. So let's imagine that we have two essential enzymes, this black enzyme here and this gray enzyme, and they're both essential. So if you were to delete this gene, you couldn't get a colony on a plate. So if we could identify an inhibitor that, again, fictionally engaged both of these targets at sort of 2/3 level inhibition, let's say that when you inhibit this black target, the bacteria die, and you inhibit this gray target, the bacteria live. So despite the fact that both of these enzymes are essential for bacterial growth, they are not the same. What we would say is that this black target is more vulnerable than this gray target. And I'll define what I mean by that in the next slide. And so this is what we became very interested in trying to understand, thinking about actual qualities of the target, and can we in principle identify the most vulnerable targets in TB, and might those be good starting points for target-based drug discovery campaigns? So what do I mean by vulnerability? So the way we define this is vulnerability is a continuous variable that relates the magnitude of gene inhibition with the resulting impact on cell fitness. So let's imagine we have 4 genes in this bacteria, 1 non-essential gene and 3 different essential genes. And the goal of this vulnerability approach is to generate what we call these expression fitness relationships. In this scenario, we're sort of a standard all or nothing genetic approach like gene deletion or most forms of transposon mutagenesis. You really have access to these 2 windows, right? You've got the wild type expression level, wild type phenotype, you've got the null phenotype. And here you can very clearly distinguish this non-essential gene in blue from the 3 different essential genes. But it's very difficult to distinguish the 3 essential genes from each other, at least from a fitness perspective. So the goal of this vulnerability approach is to generate the complete picture, generate this expression fitness relationship for these genes, because in doing so, you can see that these 3 essential genes are not the same. This purple gene, for example, requires less inhibition in order to impose the same fitness cost as this green gene. We would say this purple gene is more vulnerable than this green gene. So that's the idea is behind this vulnerability approach, is to systematically titrate gene expression, build these expression fitness relationships, and move beyond this sort of overly simplistic binary essential versus non-essential genetic view of the world into this continuous trait that we call vulnerability. And the way that we do this is by using this method called CRISPR interference. Hopefully most of you are familiar with this. I got ahead of myself. So this work, it was published a couple of years ago, and it was led by now a former PhD student, Barbara Bosch, a man who might be familiar to some of you in the audience, Michael De Jesus, who developed some of the leading statistical methods to analyze transposon sequencing data, and my dear friend and colleague at Cornell, Dirk Schnappinger. So the beauty of CRISPR interference is it's really simple. So you take Cas9, normally a DNA nuclease, you inactivate the nucleus domain so it's catalytically dead or decas9. You give it a single guide RNA or a guide RNA. It forms a ribonuclear protein complex that then scans the genome, and where the sgRNA base pairs to its target site, Cas9 sits down and it blocks RNA polymerase from transcribing that gene. So this is a transcriptional interference mechanism. We use a different Cas9 enzyme than what most people use. Most people use Cas9 derived from strep pyogenes. I won't get into the reasons why, but we use a Cas9 enzyme derived from Streptococcus thermophilus, STH1. This is anecdotally the Cas9 enzyme derived from like the original Dannon yogurt guys. So it's a lot of fun. One of the big differences between this STH1 Cas9 enzyme and pyogenes is that this STH1 enzyme recognizes a more complex PAM sequence. The PAM, or protospacer adjacent motif, is this sequence within the DNA target that licenses this R loop formation. It's the obligate first step in target recognition. And what we recognized pretty early on is that this STH1 enzyme can actually recognize so-called non-canonical PAMs at the cost of the magnitude of target knockdown. So here we're targeting a Rhinella luciferase reporter in our model mycobacteria, Mycobacterium smegmatis. And simply by targeting different PAM sequences within that gene, we can tune the level of knockdown over about a 200-fold range. So at this point, we had a very robust system. We can induce very high level knockdown of nearly any gene in the TB genome. We thought we could predictably tune the level of knockdown by targeting different PAMs, and another trick that I'll show later. And so to quantify vulnerability for almost all TB genes, what we needed to do next is turn this into a genome scale sort of functional genomics approach. So what Barbara did is she cloned an array of about 97,000 different sgRNAs. So for non-essential genes, we just targeted them with the strongest predicted guides. For genes that are essential for in vitro growth, we really carpet bomb them with sgRNAs. So sgRNAs we thought would be really strong, sort of in the middle, and very weak. Print these basically using a microarray, clone these into a CRISPR interference plasmid, transform this into TB. And now what you have is you have a pool of Mycobacterium tuberculosis, where each individual bacillus has a single CRISPR interference plasmid with a single sgRNA targeting a single gene. You can then perform a competitive growth experiment. So our CRISPR interference system, so expression of Cas9 and the sgRNA are induced by ATC, or anhydrotetracycline. So minus ATC, CRISPR interference is off. These bacteria are basically wild type. Plus ATC, CRISPR interference is on. Grow this over about 30 generations. Harvest aliquots of the library every 2.5 to 5 generations, and then count sgRNAs by deep sequencing. And the beauty is that these sgRNAs are basically molecular barcodes, right? They tell you what gene you're targeting and where in that gene you are targeting. And then comes the fun part. How do you quantify gene vulnerability? And at the time, we reasoned that there were at least 3 things we wanted to know for every gene. So first, what is the maximum fitness cost? So if we target that gene with a predicted very strong guide, say 200-fold knockdown, how quickly do those sgRNAs deplete from the library? 2, what's the sensitivity to partial knockdown? So if we target that gene with sort of a medium strength guide, what kind of fitness cost does that impose? And then 3, what's the phenotypic lag? So how long between when we add ATC to when we start to see a phenotype? So a vulnerable gene has a high maximum fitness cost, it's sensitive to partial knockdown, and it has a short phenotypic lag. And an invulnerable gene is just the opposite of those 3 things. So the first thing you have to do when you develop a new genomics method is make sure that it's not a random number generator. So what we did is we asked, well, how well do gene essentiality calls defined by CRISPR-I correlate with what we know from the ground truth from transposon sequencing data? So in this heat map, every row is a gene, and these dark blue genes here are genes that when you inhibit them by CRISPR-I strongly deplete from the library. These are our essential genes by CRISPR-I. And what was encouraging to see is that the vast majority of genes that are defined as essential by transposon sequencing, right, again, our ground truth data set, are also essential by CRISPR-I and vice versa. Most genes that are non-essential by TNSeq are also not essential by CRISPR-I. There are exceptions. Some of this is real biology, some of this is artifact, but from a 30,000-foot perspective, the method is producing results that are consistent with what we know that has already been published. But we don't care about defining gene essentiality. We could already do that with existing methods. What we want to know is gene vulnerability, right? Build these expression fitness relationships. Ideally, what we would know for all 96,700 guides in that library, we would know how much knockdown that we're getting for that sgRNA, but that is not practical to actually quantify that directly. But what we reasoned is that we could computationally infer how strong these sgRNAs are, which is our proxy for the magnitude of target knockdown, by modeling their behavior in the screen. So here are three different genes, or three different sgRNAs targeting an essential gene MMPL3, a strong, medium, and weak guide. Don't worry about the math, but what Michael does is he fits just a piecewise linear regression model to these sgRNAs, and the only parameter that really matters is this beta E parameter. So that's the slope of this decay phase. So that is, in effect, the fitness cost imposed by that sgRNA. So Michael does this for all 96,700 guides in the library and then feeds the output into a very simple form of machine learning called a linear model, and asks the model, well, what features of the sgRNA predict how strong that sgRNA is, with that strength being this beta E parameter. And what the model spits out was not surprising, which was quite good in this case. So the bigger in magnitude that linear model coefficient, the more that this feature is predictive of how strong that sgRNA is, what kind of fitness cost that sgRNA imposes. The three features that matter are what PAM is the sgRNA targeting. Is it a PAM the enzyme really likes versus likes much less? We also reasoned that we could probably tune the level of knockdown by playing with the length of that sgRNA targeting sequence. So just like if you want to lower the TM of a PCR primer, you just make it shorter. Same thing works for an sgRNA. Short sgRNAs produce less knockdown than longer, and either extreme of GC content was bad. None of this was surprising, again, which is good, but what the output of the model gives us is it gives us a quantitative prediction for how strong every sgRNA is in that library. So again, that's a prediction. You have to test these predictions. So what Barbara did is she cloned 29 different sgRNAs of varying different strengths against the Ranilla luciferase reporter and asked, how well do the predictions of sgRNA strength correlate with the actual magnitude of target knockdown? And the answer was, it was actually quite good. It's not perfect, for sure. The model isn't 100% accurate, but the results are quite good. So we normalize the data. So 1 is a very strong guide, lots of knockdown in dark red. 0 is a very weak guide, very little knockdown in dark blue. So here's what the output of the screen looks like. For this essential gene MMPL3, so every line in this diagram is the behavior of a single sgRNA targeting MMPL3. In dark red, the predicted strong guides, you can see deplete quite rapidly from the library. The dark blue guides, to a first approximation, don't. And because of the scale that you can work with these sorts of approaches, you can get every sort of quantitative phenotype in between. Cleghorn is a gene that is not essential under these growth conditions, and under these conditions, again, the sgRNAs don't deplete. So again, these data, these sgRNA strength predictions are not perfect, but they pass the eyeball test. So then how do you go from sgRNA level data into these expression fitness relationships to quantify vulnerability? So what Michael did is for every gene, he fit a hill curve. And again, don't worry about the math, but here's expression on the x-axis. So again, 1 strong guide, lots of knockdown, 0 weak guide, very little knockdown, fitness on the y-axis. Just to orient you to the relevant parameters here. So beta max, this is the maximum fitness cost. So what kind of fitness cost do you impose when you target that gene with a very strong guide? M is the sensitivity to partial inhibition. So how strong of an sgRNA do you need in order to impose half max fitness cost? And then I'm not showing it here, but we also quantify this gamma parameter, which is the phenotypic lag. This is, of course, cartoon form. Here's what the actual data look like. Here's again this essential gene MMP-03. Doesn't really matter what it does for the purposes of this talk. This is the expression fitness relationship for this essential gene MMP-03. Every dot is the behavior of an sgRNA. And so what Michael does is he fits a hill curve. It's Bayesian and iterative. So in gray is a single iteration through the model, and black is the mean hill curve derived from all 10,000 iterations of the model. As you can see, there are certainly some noise to the data, but if you were going to draw the curve through the data, that's probably about where you would put it. So this is vulnerability for this essential gene MMP-03. Michael does this for all 4,000 something genes in TB. And so are all essential genes the same, or are they different? Do they look different from each other? And what was really quite nice is that, of course, they do look quite different from each other. Here are 4 different essential genes in TB. If you were to try to delete these genes, you could not get a colony on a plate. So by a standard sort of all or nothing genetic approach, they look very similar to each other, but you can see by this vulnerability or this expression fitness relationship metric, they're very different. So you have this highly vulnerable gene MMP-03 and this otherwise essential but extremely invulnerable gene MOB1 here. So not all essential genes are the same. They have very different vulnerabilities. To quantify vulnerability and sum it into a single value, which makes a lot of downstream analyses easier, what you do is you take the integral, you take the area above the curve. The bigger that area, the more vulnerable that gene is, the bigger the fitness cost associated with knocking that gene down. And when you do this for all genes or almost all genes in TB, you get something like this. So here in the Circos plot, every dot is a gene. And the closer that dot is to sort of center of the circle, the more vulnerable that gene is. And there's really just sort of 2 take-home messages from this slide here. The first is that essential genes vary widely in their vulnerability. They exist along a gradient of sensitivity to genetic inhibition, with some extremely vulnerable genes and some extremely invulnerable genes. The second sort of take-home message from this slide here is that the targets of 2 of our most potent first-line therapies, rifampicin, which targets RNA polymerase, isoniazid, which targets INHA, it's encouraging that these 2 targets are in the upper quartile most vulnerable genes, but you can see dots in red or genes that are even more vulnerable than these highly potent targets of first-line TB therapies. So what are the most vulnerable and invulnerable genes in TB? Many of them you probably didn't need to do an experiment to have guessed, right? Central dogma processes, you have to be able to replicate your chromosome, you have to turn DNA into RNA, RNA into protein. These are highly vulnerable processes. So genes that encode ribosomal proteins are extremely vulnerable. Quite interestingly, tRNA synthetases as a class are basically universally vulnerable, and this contrasts with many amino acid biosynthetic processes, which were comparatively invulnerable. So at the point at which that amino acid needs to get ligated onto a tRNA, this appears to be a sort of choke point in translation. TB is a very complex cell envelope, which you'll hear about in the next 2 talks. So mycolic acid biosynthesis, so these long chain fatty acids that make up the outer membrane of TB, this process is very vulnerable, which is potentially consistent with the large number of whole cell active inhibitors that we have that target this process. But there were lots of things that you couldn't have predicted without doing the actual experiment. I'll show you just one here and encourage you to go read the paper if you're interested. For example, the protease is oddly essential in TB. It's not essential in E. coli. It's not essential in B. subtilis, and it's highly vulnerable in TB for reasons we don't fully understand. On the flip side, I touched on this a little bit, but metabolism as a whole tended to be highly invulnerable, and we were quite excited about this, but it turns out the metabolism people discovered this many decades ago, that many metabolic enzymes are present at levels that are much higher that are needed to maintain flux through that pathway. So for example, coenzyme A biosynthesis is essential in TB, but comparatively invulnerable. Under these growth conditions, sulfur metabolism is highly invulnerable. It's very well appreciated that vulnerability is going to change as a function of the growth environment of the bacteria. It's going to change as a function of genetic background, and that's a major area of focus in the lab now. So what does this mean for target-based drug discovery in TB? Well, the idea is really, of course, quite straightforward. All else being equal, there's a lot baked into that statement, but all else being equal, if you have a limited number of targets that you can prosecute, pick a highly vulnerable target like RPOB and avoid a highly invulnerable target like, for example, peptide deformylase, with the idea being that prioritizing highly vulnerable targets and deprioritizing highly invulnerable targets might increase the chances of success for target-based drug discovery to go from something like this, where we've been historically, to where we're at now, which is kind of cross your fingers and we'll see in the next 5 to 10 years how we do. So we're part of the Gates-backed TB Drug Accelerator. We're very grateful for that. And as part of this big public-private consortia, we use things like vulnerability to actually try to pick the next targets that we're going to pursue for target-based drug discovery. So I'm very grateful to the Gates Foundation and grateful to the lab. And back to you, Val.
Thank you so much, Jeremy. I mean, it's a brilliant exposition of an incredibly powerful tool. It provides, and I can see a number of people are applauding. So this has gone down very well, not surprisingly. Your talk has provided now the ideal segue into our next talk. You've already mentioned Dirk Schnappinger, and it's just a great privilege. I've known Dirk for many, many years. He's an outstanding scientist whose work has revolutionized our ability to identify and to validate TB drug targets. Dirk joined Weill Cornell Medical College in 2001. He currently holds the position there as professor in the Department of Microbiology and Immunology. He hails from Germany, where he did his PhD at the Friedrich Alexander University of Erlangen-Nuremberg. And he worked on the repressor controlling tetracycline resistance in gram-negative bacteria. And as you'll see in his talk, the work done in his graduate studies has profoundly influenced his thinking. And he's developed tools over the years on using this kind of system to switch genes on and off. After his graduate work, he moved to the US, to the West Coast, where he worked firstly at UC Berkeley and then at Stanford with Lee Riley and Gary Skulnick. And in the Skulnick lab, he helped to adapt microarray RNA profiling to the analysis of bacterial pathogens. In fact, the work that Dirk did there has provided the TB field with some of the most profound insight into the way in which the organism adapts to the various environments encountered during host infection, including hypoxia, nitrosative stress, oxidative stress, and life inside a host cell such as a macrophage, profoundly important to the entire field. Currently, he works on and is singularly focused on developing and discovering new TB drugs. Using these tools to switch genes on and off, he has worked very closely with Jeremy, in fact, to integrate target-based and phenotypic approaches to drug discovery, and his ability to be able to inactivate genes selectively has had a profound influence not just on TB drug discovery, but also on the improvement of the efficacy of the BCG vaccine. And very recently, he and collaborators published and developed a human challenge model for TB. Dirk, it's a privilege to have you here. Over to you.
Thank you so much. That was a fantastic introduction, and it feels like it can only go downhill from here. All right, let's see. Yeah, so I would like to discuss 3 different chemical genetic tools that we use in the context of drug development. That's the validation of MTB targets with a little bit of a focus on how to do that in mice. Then how to use the platform Jeremy developed to perform mechanistic analysis of hidden lead compounds, and then a chemical genetic way of helping to discover new chemical matter via target-directed whole cell screens. So why conditional gene silencing? Most antibiotics target in vitro essential gene products and processes, which are difficult to study using traditional genetic approaches and screens, such as transposon-based methods, simply because you can use transposon-based methods to define if something is essential, but you can't study the consequences of their inactivation. And successful treatment of an infection requires targeting gene products and processes required by a pathogen to maintain an infection. And conventional mutants, such as transposon mutants or deletion mutants, often only allow one to assess the importance of a gene product for establishing an infection, but don't allow you to study the consequences of inhibiting that gene product after an infection has been established. So that's why a number of years ago, one of our goals was to develop a conditional gene silencing system that allows to efficiently suppress gene activity of MTB in vitro and during infection. And as you may have guessed from Val's introduction, we relied on the tetracycline regulated gene expression system that is part of the transposon TN10. And the most sophisticated version of that system we developed combines 2 modes of silencing. One, transcriptional repression. So in this case, we would have a gene of interest here. It's just a reporter gene, luciferase, in which we bring the gene under the transcriptional control of one of these tetracycline repressors. And then we modify the C-terminus of the encoded protein to encode a tag that by itself is not a good substrate for the CLIP proteolysis machinery, but that upon activation of a accessory factor actually allows us to deliver the encoded protein to that proteolytic machinery. So when we add tetracycline, then we have one version of the TET repressor that has been modified so that it requires tetracycline to bind to its operator. So adding of tetracycline turns transcription of the targeted gene off. And at the same time, we induce that accessory factor that I mentioned before that will deliver the encoded protein that was created before we added ATC or tetracycline to the proteolysis machinery. And the nice aspect of this system is that it achieves a large dynamic range. And the other aspect of that that's not illustrated here is that the mutants are more stable than mutants that depend on one of those mechanisms of gene silencing. And that is what allows us to, that's one of the features that allows us to use this system to test the importance of individual genes, MTB genes in mice. And this system was developed by Ji-Hyun Kim and Catherine O'Brien, who at the time were graduate students in my lab. So one of the genes for which we tested the system first was the NAD synthase, which is encoded by the NAD-E gene. And as you can see here, we infect mice with this dual control strain DUC, so 2 modes of gene silencing. We either silence from the beginning just to see what happens if we inactivate right at the point of infection, And then we can let the infection progress. And by adding doxycycline, which is a tetracycline derivative that's easier to use in mice, we can, when we switch mice from doxy-free mouse chow to doxy-containing mouse chow, then we can turn on the silencing machinery and then observe what happens to MTB during an infection when we are able to silence the targeted gene product, with the hope to somewhat predict how efficient a small molecule inhibitor of the targeted gene would be if we had one that's bioavailable in mice. And here you see the consequences in lung. There's this drastic impact on growth and survival if you deprive the bacteria of the ability to synthesize NAD, and that is not organ specific. You see the same in spleens. What we learned since tuberculosis is a very complex disease with different types of pathologies where the bacteria encounter different kinds of microenvironments, we are also interested in testing how silencing of a targeted gene affects MTB under a variety of different conditions. And here we focused on non-replicating MTB, either made non-replicating by starvation in PBS, that's on the left side, or by after adaptation to hypoxia. And in both cases, silencing of NAD has a drastic effect on survival of the bacteria. What we then later found out is that the elimination of CFUs in response to silencing of a targeted gene can predict sterilizing activity, but doesn't have to. And that became obvious when we studied two other kinds of mutants, one in which we interfere with expression of the so-called biotin protein ligase, BPL, which is required to utilize biotin and covalently attach them to biotin requiring enzymes. You see, these are three different kinds of experiments performed over, actually, fortunately, several years. We let the the infection progress, then turn the gene off by switching mice to doxycycline-containing chow, and then follow CFUs. That's the type of experiment I showed in the previous slide. The difference here is then that we asked, okay, when we now switch the mice again to doxycycline-free food, is there any relapse that we can observe? And for the BPL mutants, there's relapse is very, very rare. And this, we tested that in 3 independent infections. If you look at another mutant in which we silenced an also in vitro essential gene that's involved in redox control, then you see the initial part of the experiment looks very similar. So in both cases, silencing of the targeted gene leads to efficient elimination of CFUs in the mice. But when we switch to doxycycline-free food for the TXB mutant, we actually see frequent relapse. So that means in some cases, this elimination of CFUs actually predicts sterilizing activity. In others, it doesn't. We'd, of course, love to know this on a genome-wide basis for all essential in vivo essential MTP genes. But these, as you can see on the timeline, these are rather lengthy and resource intense experiments. So we can only do that for, at least for now, only for very few targets. So the principle here is that we use gene silencing to predict the impact of inhibiting a novel potential drug target, and then we would, of course, bias our development efforts towards those targets that have a drastic effect in vitro under a variety of conditions, and ideally lead to sterilization in mice. Okay. The next part that I want to touch on is what I mentioned before as the mechanistic analysis of hits and leads. There are, of course, a number of different approaches that one can use to identify the mechanism of action by which antibiotics or hit and lead compounds interfere with growth of Mycobacterium tuberculosis. I'm sure you're all familiar with, for example, the identification of resistant mutants and then the identification of mechanisms that are involved in resistance, which can include mutations in the target. There are a number of pattern recognition approaches where one either use RNA-seq metabolomics, morphology, or a number of other assays to classify the mechanism of action of a compound. The approach I will focus on, as that's the one that we use most in the lab, is to exploit the relationship between over and under expression of a protein and the impact on the potency of a small molecule inhibitor. So as you underexpress the direct target, you expect the potency of the inhibitor of that target to increase, the MIC to go down, and if you overexpress, you expect the opposite. I will first present data, how we did this with single strains, and then summarize how we collaborated with work that Jeremy's lab actually led on using the CRISPR-I platform to do these kinds of experiments for the underexpression part on a genome-wide basis. So the target I'm going to mention for which we developed single strains initially is MMP-03. That's one of the highly vulnerable genes that Jeremy already introduced. It's involved in cell envelope biosynthesis. The reason we were interested in this particular target is because it came up as a frequent hit in phenotypic screens, with a lot of compounds being identified that were susceptible to resistance mutations in MMP-03. And we therefore thought it's worthwhile to have a strain that allows us to interrogate MMP-03 as a target for various compounds specifically. And the way we did this, and the way we do that for all experiments where we want to do single strains, the first task is to make a mutant that with and without tetracycline growth as similarly to wild type as possible. We call these TET-on mutants. In this case, the mutant was generated by replacing the promoter, the native promoter of MMP-03 with a promoter that's susceptible by repression of the tetracycline repressor. So this strain, so with ATC, transcription of MMP-03 is turned on, and without ATC, it's turned off. We made variants in which the expression without inducer is still enough to grow, but we aim for underexpression relative to wild type. And here you see this quantified, where you see that with ATC, we actually achieve overexpression of MMP-03 relative to wild type. And in the absence of ATC, we achieve a certain level of underexpression. The overexpression is because the promoter we use in the tetracycline regulated system is stronger than the native promoter of MMP-03. And this is work that Shipra Grover has led, who at the time was a postdoc in my lab. And here you see how a compound that actually engages MMP-03 behaves in this kind of assay. Without ATC, it becomes more potent. With ATC, due to the overexpression, it's less potent than wild type. So this is the behavior you expect to see for an on-target compound that engages MMP-03. And the important part here is what at the time what we discovered here at the first time is that this combination of over and underexpression is actually quite useful, because when you underexpress MMP-03, you also make the bacteria more susceptible to compounds that don't directly engage MMP-03. With rifampicin, a good example of that, you see without ATC, rifampicin becomes more potent against the strain. And this is because rifampicin is a fairly large and complex molecule. As you silence MMP-03, you interfere with the integrity of the cell envelope. Rifampicin gets taken up better and therefore shows a higher potency in the absence of ATC. But what you don't see is that shift with ATC to the right of the MIC curve, and this is because this is not a directly engaging compound. So now, this is the platform Jeremy introduced so elegantly in the talk before. One can now use that genome-wide library of CRISPR-I mutants and expose it to a compound of interest and then ask which targets, which genes make the bacteria more or less susceptible to growth inhibition upon CRISPR-I mediated silencing of the targeted gene. And this is work that's published. As I mentioned before, this was led by Jeremy's lab with Shuchi Lee, who was a postdoc at the time, and Nick Poulton, who was a graduate student in Jeremy's lab. When we do these experiments now, we actually do them in fairly decent dose response curves. We usually use 9 concentrations, and the advantage is on the left side of this curve, on the side where you don't have a lot of growth inhibition, that's the part of the curve that's most informative to identify putative direct targets of the inhibitor being studied. And genes involved in intrinsic resistance become more obvious as you increase the level of growth inhibition. And I'm going to illustrate that in one example. This is done for an inhibitor of ATP synthase. On the upper right, you see the MIC curve from that profiling experiment. And then you see 3 concentrations for which we counted sgRNAs after exposure to the compound. And these are from left to right at increasing compound concentrations. As I mentioned, this is an inhibitor of ATP synthase, and as you expect, the genes encoding subunits of ATP synthase upon silencing decrease fitness of MTB, make the bacteria more vulnerable to inhibition of ATP synthase because there's less of that active complex that needs to be inhibited by the compound. And this becomes obvious even at very low compound concentrations. Then if we look at genes that mediate intrinsic resistance, as I mentioned before, these stand more out as you increase the level of drug pressure and increase the relative fitness advantage that resistant mutants have. And this is a well-known transcriptional regulator of clinical significance. It mediates intrinsic resistance to bedaquiline and other molecules of this class because it represses an efflux pump that decreases concentrations of bedaquiline and similar molecules in MTB. So as you silence the repressor of these efflux pumps, the efflux pumps get expressed more, you get more efflux, and therefore the bacteria are more resistant. And then if you ask how do the genes behave that encode the efflux pump, they behave pretty much like the target of bedaquiline. They are also in the upper left corner of these volcano plots. So the magnitude alone that you achieve in the level of sensitization upon silencing doesn't allow you to distinguish between direct targets and for example, efflux pumps. You need to do additional experiments to make that distinction. Ideally, one would, of course, combine this with genome-wide overexpression studies, which is something we are working on, but that is not online yet. Okay, so the last tool that I want to mention was first established by Merck, and in the field of TB, the first time this was actually demonstrated to be useful was by Valerie Mizrahi's lab. In this approach, one uses the type of hypomorphs that I mentioned before, like the MMP-03 strain, which increases sensitivity to inhibitors of MMPL3 to identify new chemical matter that has a good chance of engaging with the gene that is silenced in the screening strain. In collaboration with Ekin Johnson and Deb Hung, and this is a project that was led by Ekin and Deb Hung at the Broad. We adapted this target directed wholesale screening approach to a almost genome-wide approach for in vitro essential genes. In this case, this platform was developed before Jeremy developed the CRISPR-I system. So at the time that wasn't available. Today we would of course do this, or one would of course do that with Jeremy's system. At the time we used the controlled proteolysis system that I mentioned in the dual control strain. Here as a single control modality, just targeting the C terminus, encoding part of the gene at the degradation tag. With the degradation tag we added a barcode. We did that for several hundred in vitro essential genes. These were then grown individually, combined, and then used to screen libraries of compounds. And very similar to the way CRISPR-I libraries are analyzed now, you count barcodes by deep sequencing. And that gives a really rich data set that allows you to use the behavior of individual hypomorphs to make educated guesses, I would say, about the target of specific compounds. And you can zoom in on compounds that have a chance to engage a target that you find attractive and that you want to pursue. This is, I think, a very elegant and powerful approach. The downside is that at least, and still, it's a quite expensive assay to run due to the cost of sequencing. Yeah, and then I mentioned the first authors of each of the studies. I want to again thank the collaborators. Jeremy has been a fantastic recruitment to the area. We're really happy to have him across the street and be able to interact on a daily basis. And Deb Hung and Ekin I mentioned as having led the chemical genetic screen for new chemical matter, and we are of course grateful for funding from a variety of sources. And this work is specifically dependent and supported by the Gates Foundation, which it would not have been possible to do most of this work without that support. And I look forward to answering questions if there are any.
Thank you so much, Dirk. Again, I'm seeing many people applauding. This is just a beautiful, beautiful illustration of taking work to genome scale and really energizing the very earliest stage of hit identification and target validation. So thank you. We're going to now move to the next speaker, who is Laura Cleghorn. And again, it's a thrill for me to introduce Laura, who I've known for many years. Laura is the tuberculosis portfolio leader in the Dundee Drug Discovery Unit, otherwise known as the DDU at the University of Dundee. Laura leads an interdisciplinary team of researchers who are focused very specifically on identifying novel inhibitors that have the potential to be progressed into preclinical candidates. So Laura is a chemist. She obtained her undergraduate in chemistry from the University of Edinburgh in Scotland, and then she worked in industry as a graduate medicinal chemist before going to the University of Leeds in the UK, where she obtained a PhD in organic chemistry in the lab of Professor Ron Grigg. In 2006, just as the drug discovery unit was being established, she moved to Dundee as a medicinal chemist, and initially she worked on human African trypanosomiasis before she also got bitten by the TB bug in 2013. Very fortunate for us. She worked as in TB as part of the TB group from 2013 and became the portfolio lead, where she leads a large and complex and highly productive team. And this was accompanied in 2022 with her promotion ad hominem to reader at the University of Dundee. Welcome, Laura. We greatly look forward to what you have to say. Thank you.
Thanks, Val. Okay, today I'm going to use PKS 13 as a case study for tuberculosis drug discovery. So are the slides moving on your screen? Because they're not moving on my screen. Yes, okay. Polyketide synthase, or PKS13, as I'll refer to it throughout this talk, is an essential gene. Its role is to condense two different fatty acid chains to produce cell wall glycolic acids, and it's the sort of last step in the synthesis. It's only found in mycobacteria, and it's essential for its survival, which makes it a great thing to target in TB drug discovery. Its protein has 1,733 amino acids, and 5 of its domains are known to have function, and those are shown on your screen here today. I'm just going to on the pointer. So there are 2 acyl carrier protein domains, 1 ketosynthase domain, 1 acyl transferase domain, and 1 thioesterase domain. So by the end of today's webinar, you will have heard drug discovery approaches across several of these domains. If you are pursuing PKS13 as a target, I hope it's useful to learn the challenges we've faced. If you're more generally interested in bacterial drug discovery, I hope some insights into how we think about target-based drug discovery are useful to you. So the first 2 PKS13 inhibitors were discovered through phenotypic screening, looking at inhibition of cell growth of tuberculosis. The papers for both of these inhibitors actually came out in the same year, 2013. They're at the bottom of the screen if you'd like to refer to them. If we start with TP2, this showed a SNP converting phenylalanine in position 79 to serine in PKS13, suggesting it's the target. If we then move to the other end, the thioesterase domain, this benzofuran shown on the screen showed 2 different SNPs converting an aspartic acid at each position to an asparagine at 1607 and a glycine at 1644. This benzofuran molecule was taken on by the team that discovered it at Texas A&M to TAM16. And this was a really exciting molecule because it showed efficacy for a novel target for TB, and that doesn't happen a lot, so that's exciting. However, there was a big issue in that it had a cardiovascular toxicity flag, and this is where the Dundee team became involved in its discovery. They were funded to do lead optimizations and brought it in-house. So if we look at the overall properties of TAM16, it has good enzyme and cell activity, it has a hERG liability, and it has moderate clearance. If we then move to the left-hand side of our screen, you can see this lipophilic amine. It protonates and forms a key interaction to this asparagine. The 2 phenols on either side of the molecule also form key interactions. Now, one concern with this lipophilic amine was that it was driving the off-target cardiovascular toxicity, and this was a key focus of the optimization campaign. Can this be altered in order to overcome the risks? The project team made over 400 molecules in the series. I'm only going to focus on 2 in today's presentation. And this is just to highlight that modification of this lipophilic amine did indeed moderate that hERG activity. So if you swap out your piperidine for a 5,4 spirocycle up here, this reduces the lipophilicity of the whole molecule, and that does help to mitigate your observed hERG activity. Similarly, you can swap out your 6-membered ring for a 4-membered ring in combination with the addition of a nitrogen in your phenol, and overall, both the hERG and clearance look promising for this molecule. If we just focus on molecule 6 at the top here, we can take a look at the crystal structure that was generated. This managed to retain all the key interactions, whilst gaining an interaction with this polar oxygen, making a water-mediated bridge to tyrosine 1663. Key molecules from the series were evaluated for their macrophage activity and shown to be promising in line with the extracellular activity. 9 molecules were progressed to an initial efficacy study. The best of those were progressed to a dose-response study with our partners GSK. So in their acute efficacy study, they dose for a day, treat for 7 days, looking at 10 different doses to establish the ED99 and the ED max. Compound 6 had a good performance here and subsequently was progressed to a chronic efficacy study at Colorado State, dosing for 4 weeks after 4 weeks of infection. And again, an efficacious effect was observed. As the project progressed and was looking promising, further, more in-depth cardiotoxicity studies were carried out, and unfortunately it showed that although we were not observing HERV activity in our enzyme assay, there was a HERG risk remaining, so the series was halted. And I think the major challenge encountered by this series was that one of the key drivers for binding, that lipophilic amine, was also likely one of the key drivers for off-target cardiovascular toxicity. However, all the biological platforms had been established in-house to pursue the thioesterase domain as a target. So the team carried out a screening campaign in order to identify alternative chemical start points that optimistically would avoid that liability. So 183,000 compounds were screened from a variety of libraries that we host here in Dundee. From this single point screening, 1,500 were progressed to hit confirmation. And I wanted to give you an idea of what that hit confirmation looked like. It ran with 4 different assays. The first was dose response in the enzyme assay. This was followed up with a counter screen to remove any false positives because there were a fluorescent readout of the enzyme assay. Those that were successful were then looked at a biolayer infimetry assay for direct binding on the target. And successful compounds there were then sent to Dirk's PKS13 hypomorph strain, looking at over and under expression and checking for a shift. So once all those assays had been complete, only 2 series remained, so there's quite a lot of attrition there. The first series showed a lot of structural similarities to the benzofuran series. It retained the lipophilic amine, it retained the 2 phenyl groups, and although the central core was now a chromone, not a benzofuran, it was decided not to pursue this series, and indeed it was profiled and did show a HERG flag. We did not pursue this, but in the past few years another group did pursue this, so you can read about their efforts in the paper at the bottom of the screen. The series our team was interested in became known as the 602 series, and this was interesting because it did not contain that lipophilic amine. It did show good enzymatic potency. Surprisingly, it did not match with its MIC activity. Its HERG looked promising, although this was likely could be higher, likely limited by solubility issues, and it had a high clearance. So this was taken on for development by the team. And if we look in the middle of the screen at the overlay crystal structures of the benzofuran, TAM16, and the 602 series, you can see that they occupy very different spaces, which was a great start to the project. And indeed, the protein moves depending upon which ligand is bound. So if we move to the left-hand side, we can see in order to accommodate that benzofuran, Phe1670 has to move out, but it doesn't move for 602. In the 602 series, arginine 1563 moves out to accommodate a stacking interaction with this aromatic ring. And what was of particular interest to the team was that 602 sat very closely to a catalytic triad of serine, which meant potentially a covalent interaction could be formed. The project team made over 200 molecules in this series, systematically looking across the molecule and changing different parts. I'm only going to focus on key changes for this presentation. The dimethoxys of the original hit on the right-hand side aromatic ring could be readily changed, and this because this portion of the molecule sits in a solvent pocket. So that allows us to make a lot of changes to the physiochemical properties and also retain our potency. So the two amides that were best were this acetylene methoxy and this 4,4 spirocycle. On the left-hand side of the molecule, changes could be made, but this was typically accompanied by a loss of MIC activity. However, small groups such as this cyclopropyl alcohol were tolerated. If we go down to the bottom molecule here, you can see a nitrile has been added in this position. And the rationale for this was to try and gain that covalent interaction that I mentioned. Now, this was a great molecule, great enzyme, cell activity, no hERG activity. However, crystallography showed that this compound did not covalently bind, which was a bit disappointing as that had been a good strategy for the team. 3 molecules from this series were progressed to PK studies. However, none of them showed suitable exposure in order to justify an efficacy study, and the decision had to be made to halt this project. The team were still enthused by this, and here in Dundee we have a structural guided drug discovery coalition, and they chose PKS13 as a target to pursue with a DNA encoded library. And this was really because we were encouraged that a different binding mode had been identified. Unbeknown to the team working on it, another group, XChem and Texas A&M, also initiated a DNA encoded library screen, and they published their results last year, and their paper is at the bottom of the screen. So on the left-hand side, I have an overlay of the 2 series that I've discussed previously. I've added in the XChem ligand in maroon and the DDU ligand in green. Now, I just want to thank the DDU team here for giving me permission to present this today. It's unpublished data. What you can see is that all the ligands occupy a very similar space. The challenges faced in the DNA encoded hit that we had was optimizing the ADME properties to make the molecule suitable for in vivo evaluation was really challenging in order to get what we wanted in combination with potency, and that was really similar to what we had seen in the 602 series. Some molecules from this series also exhibited a hERG flag, and I think this might be indicative of the long channel that the molecules are sitting in, and I think it's something to be careful of if you're pursuing this target. The team were successful in achieving efficacy, but because it was moderate, a decision has been taken not to work on this series. So for the next part of the presentation, I want to swap domains. Now, a large part of what we do here in Dundee is phenotypic screening. We've screened over 1 million compounds with our partners at the NIH. In this particular screen, we were looking at a library of 225,000 compounds and identified 1 as a potent singleton. Now, following phenotypic screening, we have a range of assays that we run through in order to try and find something out about how the molecule is working. Here, a Penny-Lux assay indicated that the compound targeted the cell wall. So we sent this compound to Dirk, and he looked at his suite of cell wall hypomorphs, and this showed that 1 was targeting PKS13. So you can see the overexpression, it's less potent, underexpression, it's more potent. Resistant mutants were generated for 1, and this showed a good frequency of resistance at 10 to the -8. And these 12 mutants were found in the N-ACP and ketosynthase domain. What was of particular interest is that 1 of the mutants was very similar to that observed for TP2. There was a phenylalanine going to serine at position 79. In our mutants, it was going to leucine. And that's perhaps not unsurprising given that both compounds have this pentafluorophenyl moiety. If you look in the literature, this pentafluorophenyl is a potential covalent binder, and looking in PKS13, there is 1 cysteine at position 287, which sits within the range of all the mutants we identified. So this is potentially what the compound is targeting. Chemistry was undertaken on the hit, and this showed really good potency in the MIC activity. It had poor solubility and poor clearance. Now, the clearance was thought to be driven by that pentafluorophenyl moiety. And as the mode of action work began to read out, the team profiled TP2 in similar assays. And what was interesting was the clearance of TP2, although not in the range we wanted, was better than compound 1. So the compounds were combined, and the thought behind it was to look at 5-membered heterocyclic amines to see if those could be moderated in order to overcome some of the challenges. Compound 24 was particularly promising with good potency, good solubility, good clearance. Looking at hepatocyte clearance, it was still high. So the project team pursued a strategy of ester bioisosteres and came up with 43, which was really good across the board, and indeed was progressed to a PK study, which it showed very favourable properties for a first compound in a series. However, it was progressed to an efficacy study, and this showed no reduction in the bacterial loads, which was surprising, but more in-depth investigation into the macrophage activity showed that you really needed a high concentration over prolonged exposure to see an effect. So it's likely in the efficacy study we were not achieving that. And I think that surprised us because it hadn't been an issue with our other series, and it highlights the need to constantly be questioning your biology as you're pursuing your chemical series. So in summary, inhibitors across the PKS13 domains have been investigated. Largely, this data is available in the public domain. This has been published, and I think as we learn more about machine learning approaches, maybe there is something that be gleaned from all this information across the different groups as to how to pursue PKS13. I really wanted to highlight this suffix inhibitor that has recently been published by the Calibre and Texas A&M team. Again, you can see the literature reference at the bottom of this screen. This was also identified through phenotypic screening. The team inherited a library of sulfonyl fluorides and looked at inhibition of cell growth. They developed their hit into this compound, CMX410, which they're now pursuing as a preclinical compound. So I think that will be really exciting to monitor as it goes forward. We ourselves have retained an interest in the thioesterase domain and have been pursuing an earlier series targeting that catalytic triad, as we thought, that was something we hadn't done in the 602 series, and it remained of interest to us. So in summary, I really wanted to highlight that the work I've presented here today is a 10-year journey. Drug discovery in TB is slow, efficacy studies take a while, and I think it is important to show how a target program evolves. The challenges we faced here, I think HERG is one of the most obvious one, and I think it's something definitely to monitor if you've got a lipophilic amine, but also particularly if you're targeting PKS13. We did see limited PK exposure across our series. I don't think that is a particular issue for PKS13. You can encounter it in any project, but I think the size of our molecule has caused that issues. Identifying relevant chemical matter is a challenge for all targets. I think we can get really promiscuous targets where you can get a lot of hits. The big question is, can you make them into a new antibiotic? I think tools that really helped us in this drug discovery were access to the hypermorph strains and also structural biology, which we had to support the thioesterase projects, but we didn't have for the ACP ketosynthase. We were at the point of looking into the full protein crystallography when the project began to close. So I think that's something that's helpful. Something else I think you should consider, especially on TB, is how your target or chemotype is going to work in combination with others. And I think that's a challenge for early drug discovery projects, because often you're sitting with an early molecule, it needs optimized. Is now the time to start looking at combinations? I think it's important to consider, and a big question is, will a PKS13 contribute to a superior regimen? So it just leaves me to thank the project teams who have worked on these. There's a lot of work that goes into drug discovery. I hope this is represented by the number of names on this slide. The references for each of the papers that I've talked about are also here. I'd briefly like to thank Alice, who modified a couple of the structures for this presentation. Important to thank the Gates Foundation for funding and the Wellcome Trust for part funding the benzofuran, and thank you for this opportunity.
Yet another wonderful talk, and I think that you can all appreciate we have really been treated to some spectacular science. It's now time to go into the Q&A session. Remember that as a quick reminder, you can submit your questions as shown on this slide, and we'll do our best to respond to as many of those as possible. So without any further ado, we've got 15 minutes approximately for questions. I want to start by a question first to Jeremy from a member of the audience. Have you implemented your CRISPR-I system to assess target vulnerability in gram-negatives such as Klebsiella? If so, could you share whether the vulnerability profiles for conserved targets differ significantly? Jeremy.
Yeah, so I think that's a super important area of future research. That's not something that my lab is doing. We're quite focused on mycobacteria, given how challenging they are to work with and their medical importance. But other people for sure are doing this. With respect to conservation, I can't speak to Klebsiella, but conceptually similar approaches have been applied in Bacillus subtilis and E. coli using slightly different methods. And there are, of course, many similarities and differences in terms of what the most vulnerable and invulnerable gene sets are between TB, E. coli, and B. subtilis. Central dogma processes are universally vulnerable across all three organisms. Metabolism as a whole tends to be invulnerable, although again, there are many exceptions for different metabolic enzymes, but those in sort of broad strokes are the themes when comparing TB to E. coli and B. subtilis.
Jeremy, if I could just chip in and ask you to elaborate a bit around the question of how vulnerability might change as a function of the conditions under which the organism is being tested. And, you know, in TB, the Dirk had alluded to the multiple microenvironments. Are we getting closer to getting a handle on this?
We are getting closer. We're certainly not there yet, but it's well appreciated that vulnerability is going to change as a function of the different microenvironments that the bacteria are growing in. And that's a very active area of research. We're getting closer in vivo and different in vitro scenarios that mimic different aspects of the infectious life cycle of TB. Its vulnerability also changes as a function of genetic background. So we published some work last year on how vulnerability changes in the context of drug-resistant bacteria. These drug resistance mutations can alter the physiology of the bacteria. They can also alter the vulnerability of different genes, potentially in ways that one might be able to leverage to, for example, in theory, slow the evolution of drug resistance. So both of these areas are very active areas of research in both my lab and Dirk's lab and others.
Great, thank you. Another question from the participants on the call. I'm going to direct this one to Dirk. How does the target vulnerability or the vulnerability of specific target translate into predicting sterilizing power for chemotherapies against that target?
That's a great question, and we don't know the answer to that. My expectation would be that that there is a loose correlation between vulnerability and sterilizing capacity, but that there will be a lot of exceptions to that. So I don't think vulnerability measured in vitro will have excellent predictive power for what has sterilizing capacity. If you look at the two examples that I mentioned in the relapse experiment, those two, I don't think we could have predicted that one of them is sterilizing and one is not. And it really took the mouse experiment to find that out. As I mentioned, the downside of that experiment is if you look at the timeline, it's a 12-month experiment. If you want to convince yourself that the data are reproducible, you're talking 2 years, something like that. Making those mutants that are stable enough that they survive that experiment, because of course you have to check for the mutants or the colonies that grow up, were these escape mutants or not. So that's something that we still can't evaluate at a higher throughput, and we'd love to change that.
Great, a wonderful question and an even better answer. Laura, there's a question from you. If the PKS13 screen had to be done again, would it have been more efficient to first screen the PKS13 hypermorph before evaluating enzyme activity directly? It seems that whole cell activity is the main bottleneck. Can you comment on that?
I mean, I think the limitation there would be throughput of running that screen again. I mean, I don't know what Dirk thinks about running tens of thousands of compounds. I think that would be the limitation there.
Dirk, do you want to comment on taking?
Yeah, I can. So, yeah, I can't say if it would have been more efficient. The throughput is a challenge for some laboratories and less so for others. So my lab is not trying to do high throughput screens, and we are not equipped to analyze thousands of compounds. But labs that do that on a regular basis could do this with a hypermorph the same way they do it with wild type. The challenge there initially for a new target is to find a hypermorph that performs well in these screens. And it really requires validation of that hypermorph with a small molecule. And if you go after a new target, you don't have that to validate the strains. For PKS13, where there was chemical matter available, you could test if the hypermorph performs well, and then you could test it to what extent you get false positives and false negatives in a larger screen. I think there is flexibility in this. I'm not sure if there's one path that is perfect. I think these are modules that one can put at different places in that workflow without one being vastly superior to another. And I think there's also the added complexity of hypermorph screens is that one can find hits that act on pathways as opposed to just the individual target. And so deconvolution is always going to be a question. Dirk, your observation of the difference in relapse depending on the target. This was the TRIX-B2 versus the BPL. Do you know anything about the biology of the target that distinguishes them? I think this is a $1 million question that you've probably been scratching your head over for a long time. Yeah, and it's a question at least I cannot answer. And it's really difficult to study, right? The phase that determines relapse is possibacillary. So we can't look at the bugs in that phase and see why is one able to persist in a form that is unable to form colonies at that point in time, right? Otherwise, we would not achieve apparent sterilization and lack of CFUs at that early time points after doxycycline is switched out. What Sabina's lab has done is to use these strains to look at factors of the immune system to see how, if any of them, and to what extent of them they can control relapse. And that approach is not hampered by the fact that there is a possibacillary phase where in essence the bacteria are invisible and not available for study for us. So that is more productive and technically more straightforward than trying to understand the biology of MTB in that parsibacillary state.
Thank you. Jeremy, there's a question for you here about MMPL3. How do resistance-conferring mutations in MMPL3, and many have in fact been described, influence the druggability of this target and the robustness of its vulnerability as a target for TB?
Say that one more time, Val. How did the resistance mutations?
Resistance-conferring mutations in MMPL3 influence its druggability and its robustness as a therapeutic target. I think this really gets to the question of just how many mutations can be accommodated and what does this mean for pursuing a target?
Yeah, so it's an interesting question, and I would say to a first approximation, vulnerability is not relevant to answering that question. The available resistance mutations are going to be highly dependent on whatever molecule that you're looking at, with one potential overarching exception, in that in TB, if we look at the upper quartile most vulnerable genes and the bottom quartile most invulnerable genes, and just look at them across evolutionary scale, either just within TB or across different bacteria, what we do find is that the most vulnerable genes are under stronger purifying selection than the least vulnerable genes. And what that might suggest is their mutations might be less likely to occur in a highly vulnerable gene than a highly invulnerable gene. Of course, it's going to be very dependent on the target and the molecule, but from a 30,000-foot perspective, the most vulnerable targets might be less likely to mutate than the least vulnerable. There's going to be probably a lot of exceptions, though.
Yeah, excellent. Thank you. We have 2-3 minutes left. I'm going to throw a question to Laura here. Given your role, Laura, in leading a team of chemists, pharmacologists, and biologists, and you're based in a university where you've got, and in a unit where you've got some of those capabilities, but you also work extensively with international collaborators. Could you give the listeners to this webinar some sense of some of the complexity of these virtual drug discovery consortia and what lessons you have learned leading an interdisciplinary team in an academic, essentially an academic environment?
I mean, first of all, it's great. I mean, I think at my last count, we worked over 30 different groups, which, to be able to share science and drug discovery is very much an interdisciplinary science. I think the key thing is communication and, make sure that you have good interactions and regularly discuss your projects with your teams and have common goals in those projects that you're pursuing.
And Laura, are you integrating, AI, ML is just the rage in drug discovery. Are you confident that in the TB space, we're up with the rest of the world in terms of adoption of these new tools?
Yeah, I mean, I think it's interesting because a lot of companies want to help not-for-profit diseases, right? But I mean, we do try machine learning approaches in-house, and it is very much, it works for some things, it doesn't work for other things. It depends on the quality of your data, and I think that's something that we as a unit have been quite set on, making sure all our data sets are very robust, because you need that to build good models. But certainly, because we work on not-for-profit diseases, we don't have the money to maybe put into some of these techniques. So that is a challenge there. But I think it's something we are trying to integrate into our drug discovery projects.
Yeah, and I would say that we need to throw the best tools that we've got at this problem. Dirk, you have your hand up.
Yeah, I want to emphasize one point that Laura makes, which is, I think in TB we have benefited a lot by close interactions of scientists from very different disciplines. I think it's a privilege for us, for me, to interact with chemists and people who have real experience in drug discovery, and that has changed my perspective and what we do in the lab a lot over the more than 10 years the TB Drug Accelerator has existed. And if you look at a lot of the work that Jeremy and I presented today, to some extent, this was a reaction towards complaints from chemists that genetics are not capable of identifying good drug targets. And when you start to discuss why somebody feels like that, the vulnerability idea and measurements were a direct consequence of complaints about that genetics can achieve levels of inactivation that you might not be able to achieve with small molecules, and certainly not at the beginning of a drug development program. So that close interaction over years, and actually the criticism of our approaches by others has been hugely beneficial to us, I think.
So I think, you know, we're out of time now. So I guess it just remains for me to echo that statement, Dirk. Having worked on relatively invulnerable targets and spending years getting nowhere, but putting the work out there, which drove these kinds of new advances, it's just, we're in a totally transformed space. It just remains for me to thank our three wonderful speakers, who I think have given you a glimpse into the world of TB drug discovery at its earliest stages. It's about the vibrancy of this field, the scientific excitement of this field, and I feel huge lessons that can be applied in other areas of antimicrobial drug discovery. Thanks again, Shireen, for inviting me to moderate this discussion. Just a reminder that you can see this online, share it with your colleagues who might not have been able to attend. And so it just remains for me now to, Shireen, to hand over back to you. Thank you.
Thank you. I just wanted to say that it's been such a pleasure and an honor to have you all speak in today's webinar. So thank you, Jeremy, Dirk, and Laura, for your amazing presentations and really nice discussion afterwards. Thanks to the audience for your questions. And a big thank you, Val, for moderating the session and just helping us get this important topic out to our audience. So yeah, thank you everyone for today. I just wanted to also say a really quick word for people. You can already register for our upcoming webinar next month. It will be on October 28th. About using artificial intelligence to analyze and predict susceptibility of antimicrobials. And just, yeah, thanks to everyone for joining today. And as Valerie said, keep an eye out for our activities and make sure to share the webinar recording with your network once it's out there. Thank you, everyone. Goodbye.